and for all the previous talks. Oh, shit, yeah. Thank you so much for the invitation. It's been a really great event, first time participating, but I'm very excited about reproducibility. So it has been really nice to be part of this. And for this presentation, I have worked a lot with teaching computational skills and reproducible skills to, well, not a lot, but some to students that come from biological backgrounds. And there's people that have not even thought about what it means to be reproducible from a computational perspective. Biology students are trained to keep lab notebooks for their experiments, so they are somehow aware of the need to keep notes. But the moment that they start, and actually I should have asked first, can people hear me okay before I just get talking? Okay, thank you. So they have the idea of lab notebooks where they need to keep notes about the experiments that they do in the wet lab, but somehow the moment they move to the computer to now analyze the data that they've collected, they forget about all those skills, and then they end up with something that sadly is not reproducible, and that they cannot really get the same results when they reanalyze the exact same data. So the main message that I want to bring from this talk is that I have a set of notes that I will share the link with you, Rohan, later. If you're going to put this in YouTube, we can put the link in the chat. These are a much longer set of notes. I mean, I don't have 50 minutes to go over all of them, but they should serve as an entry point for anyone that has that wants to become more reproducible with the research. And my experience comes from biology students, but I don't think it's restricted to biology students. So anyone that wants to become more reproducible with the data analysis, this could be a good place to start. And in fact, I'm going to guide you through a little bit of the content, but it might seem trivial or even obvious for some of us that have thought about reproducibility for a while. But I really wanted to just remove the entry bar and make it accessible to anybody. So forgive me if this sounds like super obvious to some of us, but it is, in my experience, not obvious for some students that are just starting to think about reproducibility for the data analysis. So anyway, so like I said, these are a bunch of notes that are available through a GitHub page. And I'm just going to highlight a couple of them. Whoops. Oh, sorry, my AirPod fell. I'm just going to highlight a couple of them through these 15 minutes, but then again, just highlighting that both all the materials are available online, and there's also another longer YouTube video for this lecture. It's 30 minutes total that anyone can check out. And a bunch of links from people that have thought a lot more about reproducibility and computing tools. In particular, I want to point out at Carl Broman's Tools for Reproducible Research, that he has both YouTube videos and learning materials online that they're just really marvelous. It's a whole semester class, but really all materials are online for anyone to learn more about it. So I think my task here really is to point at some resources. Oh my God, sorry, my AirPod keeps falling. Pointing at some resources for anyone that feels they really want to start learning what is reproducibility, how do I start, but have never even opened a terminal in their lives. So that is the point of this tutorial. So I'm going to start just with some motivation, perhaps. Why should we even care about being reproducible? And again, Carl Broman has this quote that, well, I don't think it's his quote, but he repeats it a lot, that it says that your closest collaborator is you 6 months ago, and you don't reply to emails. So at the very least, you want to make sure that you yourself are able to understand and replicate, the steps that you had done to analyze the data. I'm not even thinking about the next step that Lars was talking about. You of course want to make sure that the moment you publish, the pipeline is reproducible as well. This very initial step, at the very least, it needs to make sense to you. So it's very important, and this is something that is difficult for students that come from biological backgrounds, is to try to do everything via code. So even apart from data cleaning, data pre-processing steps, there is a huge, I want to say your first instinct would be to do manual changes to the data. Okay, just open the spreadsheet and change things here and there. you want to try to go against that initial instinct and trying to do all, since the very beginning, the data cleaning and pre-process through scripts, because that's the only way to guarantee that first you will remember what you did because everything's in the script, and also you can reproduce it. You can reproduce it as well. And I use this phrase to name, if you see that the title is mindful programming, mindful reproducibility, I think the most important tool is really the mindset. You know, it's just saying, I really want to start this analysis with the idea of becoming reproducible. And it is indeed just a process, because no one can just be reproducible overnight, but just the intent of being reproducible will be in itself, I believe, a success. So just having that mindset. And I also like to share what I think are the 5 stages of reproducibility with at least the biology students that I have talked about. Many start thinking that because they're not computer scientists, oh my God, I'm sorry, my AirPods keep falling. Okay, there you go. We'll have to do some editing for the video. Some people think that, yeah, well, I'm not a computer scientist, why do I need to care about this? And then the moment that they start trying to become reproducible, there can be quite of some frustration, because it feels like you're doing the job twice. You are taking notes of the commands that you're running, and then you're running them. And of course, that seems like a duplicate effort when you can just run the command, or you can just click in an interface without taking the notes. So it feels like it's almost unnatural to be taking the notes and then doing them. But what they don't realize, at least no one realizes at the beginning, is that these notes are actually going to save you hours of time later on, because it is just, as I always tell students, you know, something that you're doing right now, it is very likely that you will have to redo, you know, and the struggles that you're facing right now with errors, you know, with installation, with downloading, is something that you have gained this knowledge, but you're going to forget 6 months from now. If you take good notes, even if it feels like an waste of time at the moment taking the notes, it's going to save countless hours in the future when you have to redo this process. Eventually, I would just want to reiterate that the main message that I'd like to pass to students that are just thinking about reproducibility is to try to approach this process with like slowness, you know, like a mindfulness. I love the word mindfulness, you know, just sit in front of the computer and think, okay, I'm not gonna rush through this. You have been spending years collecting this data. And like I said, my experience comes from students with biology. They go to the field, they spend years, you know, in the field collecting the data. But then the moment they sit on the computer to analyze it, they want to rush through it. They want to rush through the analysis, because that's not what they think their strength is. They really want to get to the plot. They really want to get to the result. And they want to skip the many steps in between. So one of the things that I like to highlight is just try to don't rush through the data analysis, so that you honor the process, the long process of collecting the data via a reproducible, also well thought out plan for the data analysis. So just the mindset of, okay, this is going to take however long it's going to take, you know, but I'm going to try to do it right from the beginning. And the other thing, so let me go back to some of just highlighting a few of the computer practices. Actually, I won't have time to go over everything, but there's a whole lecture on version control for people that want to learn about Git, about GitHub, like I said, without any expectation of previous knowledge from before. And right now, I think I've spoken a lot, I'm sorry. But within the last 4 minutes, I'm just going to highlight a couple of things that I think people don't think about enough when they're starting with their data analysis. And one is the organization of their folder, you know, just thinking about the names of the files. This is something that, I guess we've all been in this situation where we have files that we don't know what they refer to. We want to keep different versions, but then at one point you're rushing through the data analysis and use whatever names. There is actually a process of naming files. There are some links for some Jenny Bryan's notes on naming files in a way that you will save you time later, one, and will avoid errors when you're running things through the terminal. So some of the common messages just to avoid spaces, from punctuations, accented characters. There is this tendency to have a bunch of spaces and ampersand and everything in there. Just try to avoid that. Just keep alphanumeric characters. And also put something numeric first if the scripts need to be run in a certain order. So start with 01, 02, 03, so that you do know which is the order of the files that you're going to have to read if you want to redo them. Try to rely on text files or more fancy markdown files where you can have both text and commands. And don't just copy commands. One of the things that I have seen from students when they start being reproducible is they do open their markdown and they just copy a bunch of commands. But then later when they want to do it, they don't even know what the commands were meant to do. So just combine text, story for yourself, and then the commands. I tell people that this notebook should be kind of like a journal to yourself. Okay, I'm doing this command to get this output. This is the output that I get, and then I'm going to do this next step. So it should contain both description and also commands. And another thing that I want to point out in the last minute is even from the beginning, the way that the data is stored in spreadsheet, you also need to pay attention to that. There is a tendency in biology, like I said, that's all the experience that I have, but maybe similar in other fields, that they want the spreadsheet to be human readable. So you have your data in a way that it has colors and bold text, and there are spaces here and there, empty cells, and just for the human person that is going to see the spreadsheet. But it turns out that is not necessarily the best practice if you're going to use the spreadsheet for future analysis. So just try to think that the spreadsheet is the place where you're storing the information, but it has to be machine readable rather than human readable. It could be both, but just not using colors and different fonts and different character letters, because the computer won't be able to pick up on that. So just trying to be, so just some of the, there's actually a whole paper about data organization in spreadsheets. I put the link right here. And some of the messages that are in this paper is to try to be consistent throughout the spreadsheet in the way that you score the data, keep good names for things. So avoid spaces again, avoid special characters. Don't put empty cells, even if they look pretty. Don't try to create like a pretty image. Just always try to have your data as a rectangle, no empty cells in between. Just keep one thing per cell, make it a rectangle. Obviously, keep a data dictionary. There are countless of times where you forget what you put an acronym or you put a short description, or just a short name of something and you forget what that is. Always keep a data dictionary. And also don't do calculation in the spreadsheet. Like even there is a temptation to do some quick calculation to find out something. It's always better to use the spreadsheet as the raw data that is going to be the input read into your script later. That might be Python, might be R, whatever. And just like I said at the beginning, don't use font or anything to highlight. If you want to highlight something, for example, in a specific column, you want to highlight some of the entries, just create another column that is binary and that has, 0 means something, 1 means something else. So just from the beginning, just there are certain messages here that could ideally help you to start framing a reproducible pipeline that follows best computing practices. I'm out of time, but just like I said, all the notes are in here, and I really love to chat about these and share the experience that I've had teaching computing practices to biological students. So if anyone wants to talk with me right now or later, all my information is in the same link. So thank you. I'm happy to take questions if we have time.